Hello everyone. So this will be the final lecture on enzymes and we will talk about enzyme inhibition and applications. Okay, in your previous lecture, you have learned about enzyme kinetics, which describes the way on calculating the rate of an enzyme catalyzed reaction. And there are two important parameters which are Km and Vmax. Km describes the substrate affinity of an enzyme, while Vmax describes the theoretical maximum initial velocity of an enzyme. And then you also learn about several factors that can affect enzyme activity, such as temperature and pH. And we know that the activity of an enzyme is all depending on the structural integrity of the protein structure of an enzyme. So if an enzyme is denatured under certain conditions, then it will lose its activity altogether. Okay, here is a brain teaser from the previous lecture. Do all enzymes obey Michaelis-Menten kinetics? Well, the answer is no. If not, what are these enzymes? These are called the allosteric enzymes. And why? Well, you can look for the answer in section 8.4 in your textbook, but I'll just quickly go through the answer. The reason why allosteric enzymes do not follow Michaelis-Menten kinetics is that if you look at their activity profile, allosteric enzymes are showing this sigmoidal curve rather than the classical parabolic curve as displayed by a Michaelis-Menten kinetics calculation. Why is it so? That is because the binding of substrate into the allosteric site of an allosteric enzyme is cooperative in nature. In other words, the binding of the first substrate into the first allosteric site will promote the binding of a second substrate into a second allosteric site, and therefore you get this exponential increase of your initial velocity rather than a gradual increase of initial velocity in the case of Michaelis-Menten kinetics. So in other words, what you see here, this formula will not be accurate for calculating the velocities of the allosteric enzymes. Okay, so in the first section, we will look at different types of enzyme reactions. Although there are thousands of enzymes in nature, but all enzymes can be categorized into 6 major types based on the type of chemical reactions that they catalyze. The first type is oxidoreductases. So as the name implies, these enzymes will catalyze a redox reaction. So we know that in redox reaction, you have both oxidation and reduction happening in the same time. And in this example, A is being oxidized because it is losing its electron, while B is being reduced as it is accepting a new electron. So since both oxidation and reduction happening at the same time, this is a redox reaction. So some examples of oxidoreductases would be dehydrogenases and hydrogenases. Then you have transferases, which are enzymes that will transfer certain chemical groups, such as acyl group, amine group, or phosphate groups. So the type of chemical reaction involved here is called a single replacement because the molecule in the reactant will be replaced by a chemical group, such as a phosphate group, in the case of phosphotransferases. And then you have hydrolases, which will always function only in the presence of water molecule. And you can see that the type of reaction is known as a double replacement reaction, whereby either AB or CD must be a water molecule. And examples of hydrolases will be esterases that break down ester molecule, lipases that break down lipid molecule, and so on. And then you have lyases, which are the enzymes that catalyze the decomposition of molecules. So they decompose a bigger molecule to some smaller subunits. And in many instances, the decomposition process will introduce a new covalent bond within the product molecule. And you have isomerases, which are the enzymes that catalyze the isomerization of the reactants. So in this case, AB has become BA. So it seems that they are the same, but if you look at the structural differences in terms of spatial arrangement, there will be differences, such as D isomer turning into L isomer and so on. And last but not least, you have ligases, which are the enzymes that will catalyze the formation of a bigger molecule from simple subunits, such as synthesis. So in anabolism, ligases is very important because in anabolism, you have the synthesis of macromolecules from simple subunits. While in catabolism, hydrolases will be very important because normally it catalyzes the breakdown of complex molecules into smaller subunits. Okay, this is basically the same thing as we have just discussed, but I just want to point out that for liases, when you have got a new covalent bond formation in the product molecule, that covalent bond is normally double bonds, which means with liases, you are likely to see the addition of new double bonds in your product molecules.